Okay, hi, everybody. How are you? I am here again with the next portion from the properties of enzymes. The remaining portion today, I will present you the remaining portion. But before we proceed to our lecture, don't forget subscribing, liking, sharing these videos. Please also put your comments under this here, and let's go to our lesson, today's lesson. That is, today we shall see enzyme specificity. This is one chemical property. Enzyme specificity is a property of the enzyme that describes how restrictive the enzyme is in its choice of substrate. A completely specific enzyme would have only one substrate, of course. Yes, completely specific. Let us see some to specificity of enzymes. A little elaborate types of enzyme specificity as far as your book is concerned. You can see here bond specificity, of course, group specificity, then substrate specificity, then optical specificity, even cofactor specificity is there. These are mentioned as far as specificity of enzymes is classified, types of enzyme specificity. Let's see each one by one. You can see here about the specificity. This is a relative specificity of enzymes, where enzymes act on the reactant molecules having a similar bond. Like you can see here, for example, you can see here amylase hydrolyzes alpha 1,4 glycosidic linkage in between two glucose units in starch or glycogen. You see, two glucose molecules are joined by alpha 1,4 glycosidic linkage in starch or in glycogen. And amylase breaks this bond. So amylase is bond specific here. Even we call it alpha amylase because there is beta amylase which breaks down beta 1,4 glycosidic linkage, which is found in cellulose, the structural polysaccharide, cellule of plants, of course. The other is lipase, which can catalyze the ester bond between glycerol and fatty acids in triglycerides or in the pure lipids like fat and oil. Bond specificity, therefore, they sniff specific bond type. Group specificity, of course, the enzymes act upon the substrates that have specific functional groups. See, functional group. They are not only specific to the bond, they are also specific to the functional groups or the chemical groups around that bond. Or enzymes are specific to the type of bond and the groups that surround it, surround the bond. You can see, you can have this specific example, endopeptidases and exopeptidases. You remember, I've taught you this. These are peptidases are a group of enzymes which can break peptide bonds, the bond that holds two amino acids together. Endopeptidases cut peptide bonds at mid portion of a chain of amino acids, whereas exopeptidases can cut only coming from the tips, the left or the right tip of the polypeptide, not at the middle position. Exo from the external, the extremities, from the peripheral. And see, for example, pepsine. Pepsine hydrolyzes the peptide bond of phenylalanine, not phenylalanine only. Pepsine is endopeptidase, of course. It loves to cut amino acids from the middle of a polypeptide chain, middle section. But not only polypeptide in the middle, but those polypeptide, peptide bonds in the middle, but the peptide bonds, if the peptide bonds have amino acids which contain aromatic chemical groups, benzene ring. One such amino acid is phenylalanine, the other is tyrosine, the other is tryptophan. These are the three amino acids out of the 20 amino acids that we use for protein construction that contain the benzene ring. That's why these three amino acids are also known as aromatic amino acids. Therefore, it loves to sniff the aroma of benzene. Where is benzene smelling? Where is phenylalanine? Are you here? Are you on the middle portion of this polypeptide? Yeah. I want to cut the peptide bond you are attached to. Therefore, peptide bond that contain phenylalanine, peptide bond that contains tyrosine, peptide bond that contains tryptophan is cut here at the middle portion only. If phenylalanine, tyrosine, or tryptophan are found at the periphery from the tips, pepsin will not go and cut those bonds. Okay? Therefore, pepsin is endopeptides and specific for bonds made by aromatic amino acids in the middle portion of the polypeptide amino acid chain. Uh, Otherwise, if you go to the other example before I give you pepsin is one particular example. If you go to the exopeptidases are there. The exopeptide like the enzymes that are made by a pancreas, like chymotrypsin, and trypsin. These enzymes are enzymes which love to cut the peptide bonds from the periphery, and they love to cut amino acids that are either acidic or basic in nature. There are amino acids which are, therefore, you see, not only peptide bond, but the chemistry of the amino acids around the peptide bond is also sniffed by these enzymes, which are known as the group specific enzymes. The third group of enzymes, as far as specificity is concerned, substrate specificity, is the feature of enzymatic activity where an enzyme acts only on a particular substrate. Then you can, for example, lactase only on lactose, maltase only on maltose, amylase acts on starch but not on other molecules. You see, sucrase only on sucrose, and maltase only on maltose. You see, these are substrate specific enzymes. The other is optical specificity. This talks about enzymes specifically catalyzing one group of optical isomers. One optical specificity is also known as stereo specificity or stereo isomerism. Optical isomerism same refers to the ability of enzymes to selectively catalyze reaction involving specific optical isomers, which are also known as optical isomers, or stereoisomers are also known as enantiomers of substrate. There are two forms of optical isomers, the L form and the D form. When these optically active molecules are exposed to plane polarized light, they either rotate the light to the left, in which case the molecule is known as the L form, L isomer, or rotate the plane polarized light to the right, in which case the molecule is known as dextrorotatory, to the right, rotates to the right. So some enzymes catalyze only the L forms. Some other enzymes catalyze only the D forms. Of course, most of our enzymes, if you like, love to catalyze only one form of these isomers, the L forms. The L forms. They are specific, so the L forms, levorotatory. And example, you can see L-amino acid oxidase. This is an amino acid, and this is enzyme which oxidizes amino acids. It oxidizes only the L form of amino acids. This enzyme specifically acts on L amino acids, not on the D amino acids. So we say it is optical. It is showing optical specificity. They're demonstrating high optical specificity. It doesn't catalyze reaction involving D amino acids, highlighting its selective nature. You see, amylase, different types of amylases can hydrolyze specific glycosidic bonds in polysaccharides. For instance, alpha amylase acts on alpha 1,4 glycosidic bonds in starch and glycogen, while beta amylase acts on beta 1,4 glycosyl bonds in cellulose. This specificity is crucial for digestion of carbohydrate. You see, I have told you about this amylase specificity. This is additional uh, information. Amylase shows bond specificity here. Bond specificity here. Alpha amylase cuts alpha 1,4 glycosidic bond, whereas beta amylase cuts. Therefore, this is bond specificity. The case we have seen the first part of this enzyme specificity classification. Cofactor specificity. Here, cofactor specificity in enzymes refers to the requirement of specific non-protein molecules, which are known as cofactors. I have tried to tell you about cofactors. We shall come to this cofactor section next in elaboration. Protein molecules, specific non-protein molecules that are not only molecules, of course, they could be metal ions or salts. Protein molecules that are essential for the enzyme's catalytic activity and can influence the enzyme specificity towards substrates. Therefore, some enzymes uh, love to work with one cofactor, whereas others on other cofactor. Let us elaborate on cofactors, understanding cofactors. Cofactors are non-protein components, sorry, non-protein components, not compounds, that assist enzymes in catalyzing biochemical reactions. They can be classified into two main categories, of course. These are the inorganic cofactors, inorganic, not organic. These are typically metal ions such as zinc, magnesium, iron, and copper. They often play a role in stabilizing enzyme structure or participating directly in the catalytic process by interfering in the activity of at the active site, especially. They are required in the active site of the enzyme. Very important for catalytic activity of the enzyme, without which the enzyme will not function properly. Therefore, inorganic cofactors. The other are organic cofactors, which are known as coenzymes. As far as your book, of course, the organic cofactors. In case of organic cofactors, we also mention the prosthetic groups. Let's first talk about organic cofactors, the coenzymes. These are organic molecules often derived from vitamins that assist enzymes by transferring atoms, small molecules, or electrons during reactions. Like NAD, you know NAD. NAD is involved in many parts of the glycolytic and then in the respiratory pathways, in the glycolytic pathway, and the link reaction in Krebs cycle, and also interacts with electron transport system with complexes, electron transport system complexes. The other is FAD, flavine adenine dinucleotide. NAD is nicotinamide adenine dinucleotide. NAD is a derivative of niacin, one vitamin, B group, and also flavine adenine dinucleotide, abbreviated FAD, is derivative of vitamin B2, riboflavin. Okay, role of cofactors in enzyme activity. Of course, we have to add this point. This is what I have taken from your textbook, of course, cofactor. Cofactors in another section, I will come to that cofactor aspect of knowledge. Cofactors could be mineral ions, as I have said, the inorganic. The organics are of two types, of course, coenzymes and prosthetic groups. What makes coenzymes and prosthetic groups different is that coenzymes are derivatives of vitamins and they attach to the enzyme, the protein part, which is known as apoenzyme, only temporarily. And they can be removed by dialysis, of course, they can detach. Whereas prosthetic groups are the permanent parts of the protein. The non-protein part, which is permanent part of a protein part, is known as prosthetic group. And this prosthetic group also may contain uh, metal ions like in some enzyme, copper, like iron in case of hemoglobin, if you like. Though hemoglobin is not an enzyme, it is transporter of oxygen, of course. Let's come to role of cofactors in enzyme specificity. Cofactor specificity is crucial for enzyme function because enzyme action Many enzymes are inactive in their apoenzyme form. What is apoenzyme? The protein part of uh, the whole enzyme, holoenzyme, we call them. These enzymes are divided into two categories in their chemistry. One group, protein only enzymes. They are no any chemistry, no any chemical group. They are 100% protein made from amino acids. The other group, they have protein parts, which is known as apoenzyme, and non-protein parts, which is known as cofactor. These enzymes, which contain non-protein parts and protein parts, are known as conjugated enzymes. They are also known as holo enzymes. Holo enzymes. As far as we are talking about cofactors, we are talking about holo enzymes or conjugated enzymes, which contain apoenzyme, the protein part, and non-protein part, cofactor. Therefore, many enzymes are inactive in their apoenzyme, pure enzyme form, without cofactor. You see? Therefore, when the cofactor is attached to the apoenzyme, the whole enzyme is active. They are very important for enzyme activation. Therefore, cofactors are very important for enzyme activation. In which group of enzymes? The holoenzymes. The second, the binding of specific cofactors can activate the enzyme, allowing it to interact with substrate effectively. You see, without the cofactor, the holoenzyme is, the apoenzyme alone is inactive and meaningless. The second is substrate specificity. We require cofactor for substrate specificity. The presence of a specific cofactor can determine which substrates an enzyme can act upon. You see, we need specific cofactors for specific enzymes to function properly. For example, certain dehydrogenases require NAD+ as a cofactor to catalyze reactions involving specific substrates. Certain dehydrogenases, these are enzymes which catalyze the removal of hydrogen from a compound. They require NAD+ as a cofactor. They are specific to NAD+. The other importance is reaction mechanism. Cofactors can influence the mechanism of the reaction, such as stabilizing transition states. The transition state is the state where the substrate fits into the active site, and the transitional state means the state where the enzyme is temporarily reacting with substrate is temporarily reacting with the enzyme at the active site, so that it is made to decrease its activation energy. Short-lived transitional states. This is possible in holoenzymes because of those specific cofactors. Cofactors can influence the mechanism of the reaction, such as stabilizing transition states or facilitating the transfer of functional groups that are needed during these transitional states in catalysis process, which can further refine the enzyme specificity. You see, unless those specific cofactors are there, there is no transfer of functional groups. There is no proper transition state stabilization, and so that the product formation will not occur. Examples of cofactor specificity, you can see carbonic anhydrase. This enzyme requires zinc as cofactor to catalyze the conversion of carbon dioxide and water into carbonic acid. This is one of the busiest enzymes in your circulatory system, carbonic anhydrase. Helps the removal of carbon dioxide from your circulatory system, from your body. Therefore, the cofactor, that is mineral ion zinc, if there is zinc deficiency, the activity of carbonic anhydrase will be impaired. Without zinc, the enzyme can't function, demonstrating the importance of cofactor specificity, you see. And in the alcohol dehydrogenase, this enzyme utilizes NAD+ as a cofactor to oxidize alcohols, highlighting how specific cofactors can dictate the enzyme's substrate range and activity, you see. We need such cofactors, which are known as, in this case, coenzymes. NAD is coenzyme.